Cheetahs have long been identified as one of the most challenging species to manage in captive settings due to increased instances of disease and poor reproductive performance. Until recently, scientists were unable to explain why this was occurring. However, recent evidence suggests these problems may be related to stress. While this data makes it clear that chronic stress causes a multitude of health problems in captive cheetah populations, more effort needs to be put into developing new methods to non-invasively monitor stress in order to facilitate chronic stress prevention. In order to determine both how stress is impacting captive populations and how it can be measured, it's important to understand what occurs during the stress response. When an animal is subjected to a stressor, the hypothalamic pituitary adrenal axis is engaged. First, a signal is sent to the hypothalamus, which begins to secrete corticotropin-releasing hormone and arginine vasopressin. These hormones act on the anterior pituitary to initiate adrenocorticotropic hormone secretion. ACTH regulates the secretion of glucocorticoids from the adrenal cortex. This causes the body to become hyperalert. The most compelling evidence that captive cheetahs are suffering from chronic stress came from a study published in 2004. Scientists collected fecal samples from 20 captive cheetahs in North American facilities and 20 wild cheetahs in north-central Namibia. The results of their study showed that the adrenal cortices in captive individuals were larger, a condition known as adrenal hyperplasia, and secreted more corticoids than those of their wild counterparts, resulting in higher baseline corticoid levels. Since proving that stress is occurring, scientists are now focusing on associations between stress and the higher instances of disease in captive populations compared to their wild counterparts. The diseases of specific concern include feline infectious peritonitis, veno-occlusive disease, glomerulosclerosis, gastritis, and amyloidosis, all of which are either rare or much less severe in wild populations. FIP in particular is a relatively recent problem. The first outbreak in captive cheetah populations occurred in a North American breeding facility in 1982. Within a year, all 35 cheetahs tested positive for antibodies to the virus, and over the course of 3 years, 54% died as a result. In an effort to determine the prevalence of the disease throughout the entire captive population, researchers tested the serum of 89 captive cheetahs in North America and 16 wild cheetahs in Eastern Africa for the presence of FIP antibodies. The results showed that the antibody titers of cheetahs in the wild are significantly lower than those in captivity. Likewise, research published on the other diseases of concern indicate that stress is at the very least exacerbating, if not causing, the increased prevalence. In addition to negatively impacting the health of the animal, chronic stress is leading to poor fecundity in captive cheetah populations due to the suppression of gonadal function. In males, stress causes a decrease in testosterone and sperm production, while in females it can delay or impair ovulation. Because maintaining reproductively viable populations is a key aspect of AZA-accredited zoological institutions and captive breeding facilities, standardizing and diversifying methods of measuring stress in captive populations is of the utmost importance. Because the strength and duration of the stress response is directly related to overall health, scientists are able to measure the free glucocorticoid concentrations in order to gain a better understanding of an individual animal's well-being and likelihood of developing the common health conditions associated with chronic stress. Historically, the most common way to measure stress was through blood plasma or serum samples. Measuring the glucocorticoids present in blood samples provides an immediate and complete source of information regarding stress levels in the individual. However, the amount of hormones in circulation can rise drastically as a result of handling, restraint, or the blood draw itself. Additionally, the information is only pertinent to the exact moment the blood was taken and is therefore irrelevant when trying to assess the animal's stress levels over long periods of time. A less invasive way of monitoring stress is through the analysis of fecal samples. This method allows scientists to measure situational stress, for example, stress from introductions or treatments, as each sample is a compilation from several hours. It also allows scientists to maintain daily records regarding hormone levels in specific individuals, as they're able to collect samples from the same individual repeatedly without impacting the level of stress the animal experiences. Newer methods of measuring stress include saliva and hair analysis. Saliva analysis allows scientists to determine baseline cortisol levels, as peak glucocorticoid secretion doesn't occur until 20 to 30 minutes after the onset of a stressor, which in this case is the collection itself. There are certain limitations, however, including the difficulty of establishing a collection site within an exhibit containing multiple individuals, and the potential danger if the researcher chooses to collect by hand. Hair measurements provide the longest record of an animal's stress levels. Hair takes weeks or months to grow depending on the species, making it a valuable tool for assessing conditions of chronic stress. To collect measurements over a given period of time, the hair is clipped prior to beginning and then again when the time period is complete. This second hair clipping is what scientists measure to obtain an average glucocorticoid level from this period. Chronic stress in captive cheetah populations is a growing concern among scientists. The adrenal hyperplasia and increased secretion of glucocorticoids seen in these populations is causing chronic suppression of the immune system and subsequently making individuals more susceptible to disease. Current methods for measuring stress still have room for improvement, but scientists hope that by using a combination of techniques including blood, feces, saliva, and hair to measure prevalence of stress in these populations, we can gain a more complete picture of the stress level in individual animal experiences. With this information, they can begin implementing techniques to reduce chronic or situational stress, giving captive facilities their best chance at establishing reproductively viable populations.